This is the Biology A semester exam review. This is going to be covering all 5 units that we have covered this semester. After this review, you should be able to head over to USA Test Prep, find the semester exam that should be assigned to you already, and be able to take that test. Please make sure that you are using only your content resources to find your answers. Use the study guide and study for this test before you take the test. This information is provided from the test. It is not exact answers and exact questions. However, it's going to lead you in the right direction. So let's go ahead and get started. What environmental conditions might trigger the closing of the leaf stomata? If you remember, the leaf stomata are the small openings or small pores on the leaf, and they will open and close with environmental conditions like lack of moisture. What is the process that involves maintaining internal environments, or content internal environments? That would be homeostasis. Homeostasis is maintaining that balance. When we begin to shiver because of the temperature drop, what is that called? Is it a reflex, a stimuli, or a response? The shiver is going to be a response to a stimuli, but the correct answer is going to be response. When we shiver, we are going to be responding to that temperature drop or that environmental change. You are trying to determine if sunflowers follow the sun and set up an experiment. What would you want to track to determine if the flowers follow the sun? We would want to be tracking the direction those sunflower heads go throughout the day. So as we are looking through that sunflower, each sunflower that we plant in our experiment should be tested on where those heads are going to be facing throughout the day. In order for a conclusion to be valid, what must be kept constant? All of the variables except for the independent variable. If there's more than one variable change, the conclusion cannot be valid. So if I choose to do an experiment on the sunflower heads, I have to keep everything the same except for maybe the place that I have the plant. Maybe I have one on the outside, one on the inside by the window, and one on the inside not by a window. Everything else needs to be the same. The plant, the type of sunflower, the soil, the type of pot, how much we water it. Even those temperatures need to be the same as much as possible. While performing an experiment, why is replication and repetition important? This is talking about an experiment. So think about your experimental process. Why is replication and repetition important? How are they different from each other? So replication, it means that it can be repeated by others. If you do an experiment and you set it up and you hand me the directions, I should be able to replicate that same experiment. Repetition means more than one test. If you're going to do an experiment, you can't just do one test. You have to do multiple tests to make sure that conclusion is valid. You need to have experiments that can be repeated by others, but also that they have repetition to show that you are getting good results most of the time when trying to determine if a hypothesis is correct. What can I say about this data table in regard to the growth of mold? to determine this, I would look at this chart very carefully, and I would find out that the warmer temperatures will cause green mold to grow faster. List the 4 macromolecules and the building blocks for each of these molecules. Include the elements that they have in common. So proteins, building blocks would be amino acids. The common elements, they have carbon, hydrogen, oxygen, and nitrogen. And examples would be enzymes, hemoglobin, keratin, collagen, or amino acid chains. You can take some time, pause this video, and write down the rest of these. All organic compounds including the macromolecules, contain what element? That would be carbon. If you look back at our chart here, carbon is a common element in all 4 of these macromolecules. And since we have sugar here, and sugars are carbohydrates made out of carbon, that's where I'm getting my carbon there. List and describe the organization of the biological system, starting with the cell. Be sure you can give examples. Here we start at the bottom. Cells, smallest unit. An example would be a skin cell. Then I have the tissue, cells that are going to work together. I have muscle tissue. An organ would be tissue that works together. That would be like your stomach. Organ system would be organs that are working together. That would be like your digestive system. And then organism, organ systems that work together. That would be a human would be an organism. The molecule used by cells as a source of energy, that would be ATP, produced in the mitochondria. Is energy being produced or released in the following process? I eat a sandwich, a paramecium moves towards its prey, a wound is being repaired, kidneys eliminate waste, or pizza is digested. The 2 that would be where energy is released would be I eat a sandwich. That would be me taking in that glucose and getting energy. Pizza being digested is also going to be energy being released. The other 3 are going to be where energy is going to be used. Comparing and contrasting photosynthesis and cellular respiration. Here is the chart. Pause this video to go ahead and write some of this information down. What process do cells go through when there's low oxygen or no oxygen available? That would be anaerobic respiration. That process is called fermentation. Cell differentiation is the key process by which cells become specialized and take a specific role in organisms. Genes are expressed differently even though they contain the same DNA. Explain cell differentiation and its factors. So proteins are going to play a key role. External environments are also going to affect this. Different transcription factors are going to be associated with these cells. And then those regulatory regions of transcription factors are going to affect that expression. And the amount of light actually also helps determine gene expression in plants. What's the difference between meiosis and mitosis? Make sure you include the words somatic, gametes, 2N, 1N, fertilization, identical and unique. Here is what you should come up with. Explain the relationship between chromosome, gene, and allele. How are the traits different? Chromosomes are going to be condensed DNA, where a gene is going to be a segment of that DNA, and alleles are going to be a specific trait that is expressed, and traits are inherited from our parents, half from mom, half from dad. How are RNA and DNA similar? How are they different? Here's a good chart. Make sure that you write this fully down. What is the relationship between DNA, RNA, amino acids, and proteins? DNA calls for specific amino acids which are assembled into proteins. This is a universal code based on 4 nucleotides. RNA specifies the same amino acid and all codons for the same amino acid, and this is happening in all organisms. Describe the process of protein synthesis using these specific terms. Here's what you should have. If you have any questions about this exam coming up, please make sure that you are contacting me before the exam. The exam is due by Thursday at 11:59 p.m. If your exam is not done by that time, you will get a 0 on it and there is no chance to make up that 0. Please make sure that it is taken on time. Again, questions, make sure that you ask me through email. I can set up sessions to be able to go over this information. Good luck studying, and I hope you guys feel confident enough that you know this information. Have a great rest of your day.